What we really want to do is that using these data and then try to predict what is actually happening inside the body if doctors inject this drug into this patient. What is the likelihood of success of each drug and each drug combination?
Welcome to Target Cancer Podcast. My name is Dr. Sanjay Jadeja. I'm a hematologist and medical oncologist, also known as the OncDoc on social and news media. And today I'm excited to talk about something that we've talked about a couple of times before. I keep saying, and we kind of talk about precision oncology care. It's like, okay, we know what things have been done and what we generally use in a general sense on a general tumor, a general person. But what about the specific tumor and the specific person and the specific color of their skin, etc.? But the reason I'm excited about this episode is for two. And I'm going to save the second one for a second, for a little later. But the first reason is because we haven't really talked about precision as it applies to things that are very important to patients and caregivers, and that's the chance of a recurrence. In general, when I'm practicing, I'm giving a general, you have about a 30% chance of it coming back, and I have some metrics, maybe, with breast cancer, we have mammoprint and a couple of things that look a little more closely at features that we say yes or no, higher or lower chance. But for the most part, colon cancer and lung cancer, etc., we're just talking so generally. So I'm excited to talk a little bit about a still very real process, which is after the curative intent, cancer doesn't go away. And of course, I can't begin to appreciate how much it's on the mind of a loved one and caregiver. And then secondly, and this is the punchline, we're going to talk about how this initially started, this whole process of precision and how it's done on animals. And I love that because a person that's doing something for dogs and have put their life into it and the loved ones has to be a good person. And in our pre-call, I can already appreciate that from Dr. Seung-Won Lim. This gentleman has put a lot of time into translational medicine, bioengineering. He got his PhD at Stanford University. He was at Berkeley prior. And his passion somehow has pivoted to, one, recognizing the tremendous lapse of all this techie stuff that he's doing and how we're delivering care in this country. And then also how we can do it better. And without further ado, let's get into it. We are so lucky and fortunate and grateful for you being here, Dr. Lim.
Great. Thanks so much for your great introduction. And then it's my great honor to be here to talk to you, Dr. Sanjay. And I'm also so very excited to share our story and then why and how I got to here and what our company in Premed does for animals and also for humans.
Yeah. And I think to give some context, for anyone listening, I want them to appreciate or recognize the fact, and we've said it many times before, is you have a guideline regimen. Now, these regimens are built on studies most of the time that are years in the past. The thing that we've learned over time is, and I guess somewhat more recently, is combination therapy seems to be a good thing. And there are many reasons for this, but one of them is simply, if you can attack something in 3 or 4 different ways, that's kind of broadening your net on hopefully effectively killing every cancer cell that's in the body at the time of a diagnosis. So multiple combination regimens is something that people have gotten excited about for the last 5 or 10 years. Number 2, there's this concept which we're going to get into of synergistic effect or synergy. And it doesn't mean additive, where it's like 2 + 2 = 4. But it's like, hey, if I give one drug and it gives me a value of 2, and then I do the next one and it gives me a value of 3, but if I do them together, instead of 5, I get 9, then does it make sense to do them together because I can actually get more value from the product itself, the anti-cancer therapy in this case. And number 2, you're exposing the body for a shorter duration of time to the side effects we know that happen, invariably with chemotherapy and other anti-cancer agents that aren't necessarily traditional chemo. So with that, it seems like that's where things started for you and I believe your friend, where y'all were like, how do we make sure that if we're going to do this, number one, are we really making sure this is the right match, a right pair, a right combination to get the maximum efficacy and bring out the best version, right? If you get married, some people like, my friends, I don't get this personal, but they're like, Sanjay, I think I want to get married to this, that. I'm like, look, if they bring the better version of you out that you are by yourself, then that's a win, right? And in the same way, not to reduce the seriousness, but same with therapies. If you can get a better version of something by combining it, that's valuable. and especially if there's a finite period of time that you're delivering this care. And if I understand it correctly, that's what you wanted to assess. Number one, can we make things better? But number two, can we ensure that it's going to work on the individual specifically?
Yeah. So it goes back to like my PhD years. That when I was in the second year at Stanford PhD, I attended the seminar with like all the speakers were Stanford Medical School professors, and then one speaker actually mentioned that it was back to 2012, and then that speaker actually mentioned that combination chemotherapy was better than the single agent therapy, which sounds like a common sense, but it actually hit me really hard. And then what he really emphasized was that, well, I mean, but combination chemotherapy, if you find out the certain combination for a certain patient, and that even if it worked great, if you see the similar patient next time, it is not guaranteed that that combination exactly worked for the similar patient, because every patient has a different response. And then that was actually hit me really hard, because back that time I was trying to find out, how we can help the current patient. I was working in a pharma company and then drug development side, which is for future patients. But I was so obsessed to find out the way to improve the current patient's treatment improvement, because we already have like 200 plus FDA approved drugs out there. And I believe that we are not utilizing the what we already have at the maximum level. So I did not have any kind of idea how to do that, but that professor actually mentioned that about that. And then I just ran to the speaker and right after his talk, and this is so cool. So if we can find out the certain combination for individual cancer patients, would that be helpful? And then he said, yeah, of course, but how can you actually mix and match all these different, like all these kind of permutations? There are too many drugs out there, and then you have to test all different drug doses and all these kind of things. And then, well, if I can do that, are you going to use it? And then he said, bring it to me. So that was the moment that we found out that like there is a huge clinical needs for this kind of, there is people understand that, specific, individualized, customized combination chemotherapy is useful, but there was no way to actually try to mix all these drugs and test in a high throughput way. So that's how my co-founder and my friends and I started to build this kind of high throughput way of mixing and match and test the drugs on patients' own cancer cells. So that's
amazing. Yeah. So it's like you were one point in one moment you were excited, like, oh, we have multiple agents at once that can do better. Great. This is like, a big revolution, a big innovation, and we're going to do better. And in the same breath, it was like, but even though it can do really well, it cannot work at all. And you're like, whoa, that's a big problem. So you're trying to fill that gap on saying, okay, how do we take the gains that we know on delivering care better, but how can we make sure that it's now delivered to the right person and that they receive this? Because there is also an increased intensity to side effects. And there's a reserve that eventually kind of goes, that gets deplete, that you can't even tolerate a therapy that if it comes out a year from now and it's great, but your marrow, can't handle it. So that sounds like a tall order. I hope one day I have the confidence in myself and the gall to say, okay, let's do better. I'm going to fix it and do what you sought out to do. So I would love to know, how did you do that? I'm assuming it leverages to some degree, a high amount of deep tech. And did you go to dogs first intending for the goal of humans and that was a means to an end, or did you also just want to help dogs?
Yeah, great. So we talked about the high throughput way of the combining the drugs and the mix and match, and then apply that drugs or dispensing that drug onto the microwave plate onto the patient cancer cells very precisely and accurately. That's one technology, but another big problem to solve was that we really want to get the patient's cancer cells and then test outside of the body, because usually for that, we really appreciate medical oncologists try the best to find out the most important drug, most effective drug, highly likely to be an effective drug, but there should be a wait and see, inject and then wait and see process. So we want to try to avoid that and then trying to find out the good drug upfront and then trying to avoid the non-effective drugs. So to do that, we need the patient's live cancer cells outside of the body and then test a bunch of different drugs and the drug combinations on outside of the body. So there are two things. One is what we just described, how we can all this kind of, the mix and match the drugs. And then two, how we can actually keep the cancer cells alive by mimicking the tumor microenvironment inside the body. So that's the second thing. And then we fortunately have our special like proprietary media that keeps especially the blood cancer cells outside of the body, very similar to what is actually happening inside the body. So we have what technology. One, we can print the drugs onto these live cancer cells, and two, we can keep the patient's own cancer cells outside. So combine these two together, we can generate a lot of like extensive amount of data out of this patient's live cell plus drug, the combination. And then this generates extensive amount of measurements. And then what we really want to do is that using these data and then try to predict what is actually happening inside the body if doctors inject this drug into this patient or this drug into this patient. What is the likelihood of success of each drug and the drug combination? To do that, we have to come up with a predictive like mathematical equation for the prediction. Human brain can do that, but we are living in an era of artificial intelligence, and then we have a beautiful algorithm called machine learning. So we just trying to combine the machine learning, bring the machine learning concept into this for the analysis and then make a prediction, better and more accurate prediction utilizing these measurements. So that's why we are using, we built this live cell plus AI platform together. And then it was very natural for me and co-founders start for a human application because we both studied at Stanford and then Stanford Medical School. The professors were our collaborators, and then we all worked at the human pharma and the human cancer research. So it was very natural for us to start with a human, and then soon after we started this technology development, we realized that it's super hard if you're not a Stanford student. If you graduated out of, academia and then becoming, for-profit, company people, it's really hard to get the clinical samples and patient information out for us. So it was very shocked to me because I know where this kind of, live cell stock frozen into which freezer and then where the second box from the bottom. But the professor was definitely correct. I mean, he said, well, you're not a student anymore. We cannot give it for free or just for the research purpose. You should go through all the paperwork and then get the patient consent form signed and then proper, IRB review, all these kind of things. Okay. How long does it take? And then it takes around like 6 to 8 months. Okay. And then we only had the $3,000 in our bank account. And so our two investors, they said, like, if you can show us at least one or two real human patients' data, we're going to invest. But it didn't happen for 7 months. So that's where we, realized that, this is why there are not many companies out there who are working on live cell based this precision medicine. Then it was very fortunate for us that we were at the startup incubator, and then our neighborhood company was developing, software that manages the electronic medical record of the veterinary hospitals. And then,
no way.
Yeah, I did that. Honestly, it's shame on me, but I did not know that. Actually, dogs or cats get cancer. So I asked the CEO, and he was sitting right next to me. And, hey, do dogs get cancer? And then he he saw me really weirdly, and then, yeah, of course. Okay, wow, okay. Are there many cancer patients in animals? And yes, there are a lot more patients than humans. Really? And then do we actually do the cancer treatment? for animals. Well, he said, like, that's what my mom does. Okay, all right. I just came back to my laptop and just Google search, and there was no company doing a precision medicine for companion animals. So, and then the market was huge, and then the regulatory pathway definitely a lot simplified than the human side. So after like 40 minutes of research online, and then, okay, let's save our dogs first. That was the moment that, like, we changed our market pivot from human space to the veterinary space. And then it was the right decision, because it's very sad that dogs live a shorter lifetime than human. It's like around like 1.7. But at the same time, the tumor progression is really faster than the human side. That means that their drug response is significantly faster than human. For example, if we provide our prediction reports to the human hospital, we can validate our prediction was correct or not after at least 2 or 3 months later. But for dogs, next week. So we can build this kind of AI, developing AI, and they're validating it and then improving it. This kind of cycles we can actually did a lot faster than the human space. So that was definitely a business perspective, the benefits, but also it is a very advantageous for scientific perspective for the technology development. So, but we did not lose our, hope to expand to the human side. That's what we're doing.
That is such an interesting story. Funny and not funny about not knowing that those animals get cancers. I know, like, some are more notorious than others. Boxers, I think, unfortunately, get a good degree of like stomach cancers, and you probably know all that better than I do at this point. But what an interestingly fun fact, like elephants actually get a much lower incidence of cancer because they have like 8 or 12 TP53s, which is our our kind of regulator to make sure that something isn't faulty. They have so many. So if you have a default in your body, it gets very challenging because one of the main stops on cancer and tumor growth is thwarted or not functioning correctly. But with all that said, I love what you're doing in the sense of the tumor microenvironment, because you may hear, wait, why are we already doing that? Why don't you just take a cancer cell and just throw some treatment on it and see if it dies or not? And it's, one, not that easy. I think cancer cells live for like 3 or 4 days outside of your body. But number two, and I didn't really appreciate this, I think, until later in training, or I dare say even until early practice, on how important your immune system and the cascade of events that occur at the moment of that injury to the tumor and all of the signaling that occurs, it is just as relevant. And that is one of the main of several pillars of importance on precision medicine to me, because it matters on what's happening. Are you somebody that's on, immunosuppressive therapy for autoimmune disease? I mean, that's a whole different scenario than somebody that's not, or somebody's on steroids chronically. Like, even I would think, and I don't know this, but even like steroids all the time for pulmonary disease, like that's going to bring down certain types of white blood cells, which we know is important at the time of treatment. So first of all, how are you preserving the tumor microenvironment without giving away the entire secret sauce, I guess? And I'm sure that's a long podcast.
All the details are definitely, we keep it as a trade secret, like Coca-Cola strategy. But when I developed this kind of the special media, first of all, what I did was trying to divide the historical publication into the successful, which means that showed a good correlation between the outside of the body data and then what is actually happening inside. So there are, this kind of take out the tumor and then test a bunch of different drugs on it as a drug sensitivity assay. This was a very hot topic in 1990s. And then so many cancer researchers in academia and the pharma companies, they worked on it. And then they published a lot of paper. So insurance company actually, Aetna or Kaiser Permanente, if you go there, you can actually see the in vitro tumor sensitivity assay, something like that. And that it listed up all the publications there. What I did was divide into the successful cases and then less successful cases. And then successful cases, you can actually see that the hematology malignancy, which is blood cancers, they typically have a better correlation. the outside of the body data and inside the body data. Because, as you mentioned, it's very important. The tumor microenvironment is very important. Not only the tumor itself. So if we can divide into the blood cancer and the solid cancer, blood cancers, those are suspension cells. It's floating around in bone marrow or, in the lymph nodes or in the blood. Solid tumors like, gastrointestinal tumors or like liver cancer, these are all like stick to the nearby tissues. So it's really it's a lot more difficult to mimic the tumor microenvironment from the outside of the body. We need the other adjacent tissue samples, but blood cancers, it's relatively easier. And then because it's a floating cancer suspension tumor cells, we can actually just more focusing on the tumor cells itself. But at the same time, but still the tumor microenvironment is very important. So what we do, our strategy is that we take out the tumor cells, and then our media preserves the immune cells with the cancer cells together. So we don't trying to completely isolate the tumor cells, but also it's that there is a natural combination with the immune cells and then other cells out there. So we actually kind of co-culture the cancer cells with the natural microenvironment. So that's one of our strategy.
We pardon this interruption real quick. If you're enjoying this podcast or find it valuable for what we discuss and the education and how people see and think about cancer in general, we would very much appreciate a like, subscribe, and especially a share so we can bring that information as maximally and broadly as possible. Thank you so much for listening. It's funny, I joke, but it's kind of like if you have the entire city or town, but when you go to the post office or the DMV, it's like that is somewhat of a smaller size, but example of the proportion of everything else in town, right? It's like everyone has to go to the DMV or to the post, for a certain reason. So you're preserving that and then subjecting that area into seeing what the responses look like. And I think that makes a lot of sense, number one. But number two, blood cancers, usually anything that replicates fast responds quicker to something that affects the replication process, which is how a lot of the chemotherapies work. But what's very interesting is I didn't know that for whatever reason, the responses and the, cytoreduction, quote unquote, or basically killing the tumor, that activity is accelerated in dogs. I don't know about all animals, or at least dogs. That to me is very fascinating. And I'm sure there's some kind of evolutionary, theory on why or how that has to be the case. Maybe it's just more fastidious. Maybe it's their diet. Who knows? But you're doing all of that. So then you're able to kind of get an idea of what the combinations matter and don't. And anyone listening, I want them to know this is not a new thing as far as having an idea or assessment of something's sensitivity. We've been at the limits, humble limits of our brains doing that for a long time. If you look at a path report or your path report and you see something called FISH or cytogenetics or, molecular features, all of those things are observations. Someone said, I think this has a better response or doesn't, or this makes it sensitive to lenalidomide, where there's a certain mutation where you want to make sure they get that, We've been doing that, but again, to the limitations of the human brain, and then also to somebody thinking about it and then coordinating it. You're let's do that, but all of it. And let's just make sure, and let's use extremely computational, logarithmically to 10 to the gazillionth amount of processing and observational behavior assessment with machine learning to see, let's do way more. Let's do way more than the ones that's just, this cytogenetic is off, and it gives you a couple of buckets. But in an ideal world, you would have 100 or 1,000 buckets. Right. You would if it matters, And what I saw is you're not just considering the tumor microenvironment, two, the tumor, but also, and I say this a lot, the medications that they're on, they've received, something that they may have a progression on previously, obviously tells you that something's changed. So not only are you saying that it's not sensitive to it, right? Say you give platinum therapy and you're it's not sensitive. But the reason we all need to share our data, in my opinion, is that way we can know what that thing is that makes it not sensitive to platinum. If it was something that came out after giving someone treatment, would it be valuable to you and to me and our loved ones if we knew that mutation was present prior to getting that first-line therapy, which, by the way, today is how the guidelines work. You just have to get it. Like, unless there's these couple of, thin scrapings of knowing, oh, you can bypass this one because we know. We want to have a whole bunch of those options, right?
Exactly.
And that's what I think is valuable. It's not just what you're sensitive to, but as a whole for humankind and animal kind, I can say for once in this podcast, it's also valuable to know what doesn't work, and that way we don't have to give it even in the frontline setting. But if we have a moment, I would love to hear a little bit, because we've never talked about this, on the whole risk of recurrence. So right now, like I said at the beginning, we have these buckets of kind of like, oh, you had a couple of lymph nodes, so it's this percentage, or it's triple negative breast cancer, or it's whatever it is, high risk features on the lymphoma. But when we look, for the most part, our intervals for like evaluating patients for recurrence, one, are kind of shooting in the dark, and two, they're still pretty standardized. It's like every 3 months you repeat a scan, or every 3 months you get blood work, or maybe 3 to 6 months. I mean, that's a twofold difference. Like, it's like 3 months to 6 months is what our guidelines say. And partly because we don't know when or how or what features make it more likely to be around 3 or 6. And my heart hurts for this situation, because my patients that are in survivorship or in observation, surveillance, they're like, that's it, 3 months, 6 months. I'm like, dude, I know it sounds terrible. I'm like, it's just, I agree. I agree that it's what, that these things need to be defined more. And that is something that was the reason I was like, yes, we need someone here, because we don't talk about how the precision medicine concept also applies to prompt surveillance or prompt recognition of a recurrence, surveillance, more defined roles, and ultimately peace of mind for how often or infrequent something needs to be checked.
Okay.
And I think, or if you can get into it, how are you assessing that? Is it molecular features? Is it, the duration of how long it took to see a complete response? Is it their pharmacogenomics? Is it their immune system? How did y'all come across that? And are y'all looking at maybe hopefully one day defining how frequently or infrequently we need to check for things?
Yeah, great question. So I want to emphasize that what we have to do as all the precision medicine companies or institutions out there, any kind of researchers, we have to consider ourselves are not the decision makers. We are developing a tool to help the oncologist to make a better decision. So that means that we have to create and develop the technology that doctors want, because, clinicians, oncologists, they are the people who actually sees patient and then actually inject it and then administer the drugs. So whenever we actually develop our new technology, we talk to the clinicians and then listen to what they actually want to see. Every cancer type has it seems like we realized that every cancer type has a different needs. So I can actually give you a good example for animal space, for veterinary oncologists, especially for canine lymphoma patients, dog lymphoma patients. The CHOP is CHOP. These are the gold standard chemotherapy. So we provide, the doctors wants to see that cycle 1 and cycle 2, what is the probability of achieving complete remission? Because those are the early cycle response. And then they want to see that progression-free survival, which means that how long does it take to actually see the relapse? So that's what they want to see. So we actually develop our the models for that. What is the likelihood of achieving CR in the first 2 cycles? And then once it achieves the CR, what is the likelihood, how this patient will be categorized into whether it's a high risk for CHOP, which means that shorter progression-free survival, or low risk for the longer progression-free survival. So those are very well accepted to the veterinary oncologists, so that they can actually see that whether this patient will be beneficial for this gold standard chemotherapy or not. But for human side, especially for the human multiple myeloma patient, they want to see that overall survival. There are 3 different, the treatment regimen for VMP, 3 different drugs, VMP and then RD, or VRD. Those are the 3 different regimens that are covered by insurance companies. And then what we want to provide is based on doctors' the need. They want to see that whether this patient will be beneficial in terms of overall survival, favorable for VMP, RD, or VRD. So we can actually categorize into this patient into low or high, the risk stratification. So it depends. So we can actually do this kind of like, as long as we have the right data set, we can develop this, and then we can satisfy the doctors. It's not guaranteed, but we can try our best to provide those kind of like prediction and the visualization as survivor curves and then the bar graph, all these kind of things. How we can get that? We definitely, as you emphasize, we need the data. How we can get this point? We have to work with the oncologist starting from the scratch and then all the way towards the patient's treatment journey. So, for example, for the veterinary hospitals, every 3 or 4 months after we provide our reports, we approach the doctors and the clinic and then ask for the patient follow-up chart. So what kind of drugs did you actually inject? And then what was the clinical outcome? The medical chart actually has all the details about the clinical outcomes. And then based on that, we can validate our prediction was correct or wrong. And then it just like add that into our training model and then retrain the model and then increase prediction accuracy. For human side, we do the same thing. As long as the doctors are very, passionate about working with us, they provide us the verbally or some kind of the written, not the entire medical charts, of course, but they can, abstract the right information, necessary information, and let us know that what is how this patient was treated, and what was the clinical outcome, and then unfortunately this patient was relapsed when, and then what was the second the chemotherapeutics, all these kind of things. So it's very exciting. It's not easy, but it's very exciting and a very important journey that working very closely with oncologists.
That's very reassuring and refreshing. And I want to explain why to anyone that's listening, because it's a sobering fact, especially when it comes to myeloma, because you mentioned that a couple of times. The way we are guided mostly, outside of whether they can get a transplant or not, which even that, people are a little, auto transplants for myeloma, but that's a whole different topic. But the way we are guided for the most part is sad. And it's simply the side effect profile and toxicity of the regimens. It's like we are being asked to say, what can they handle? And can they or can't they handle it? And if they can't, then maybe consider this one. Think about that for a second. It makes sense if you know with certainty that the one that's the hardest to handle certainly means the best overall survival. Sure. Then that's a one, a size fits all. But that's not the case. And we know that. And so instead, I'm dictating on their age, their functional status, their fitness, their side effect profile. Do they have neuropathy? Do I want to make it a lot worse? But what you're doing is let's do something more. Let's see if that regimen that's more toxic actually equates or is an equivalent to survival. And that's so just what I want to say is fair. That's just a fair thing to do to patients is do you need the most toxic even if you can tolerate it? Now, this concept of, especially when it comes to blood cancers, of seeing, and I want to make sure I understood you correctly, of seeing how fast something responds is not a new one. Because when we have CLL, we have intervals, and this has been for decades, where it's okay, what happened in 3 months, 6 months? Is it partial? Is it complete? Is it astringent? We have all these terms and these very, objective kind of intervals. We also do the same with myeloma. Well, did it drop by this amount of grams per deciliter? Blah, blah, blah. So I keep saying that we're doing this a while because you're taking all the things we're doing, but you're just making it RoboCop, Tech 3.0, blue pill, use your analogy from whatever is your favorite movie. And that's what you're doing. We already do this. Let's just do it a lot, better. One, if we have the data and so many real life cases and examples. And two, for the next person, sadly, that is facing this next week with the diagnosis, can we do them better? We just do them better than what we have to offer them. So you take the CHOP. So I don't know if it's 4 cycles in dogs, but for us, it's 4 to 6 cycles. Is it 2 cycles total in dogs that they do anyway? or are they actually stopping therapy early and sparing them the additional 2 to 4 cycles because there's a good chance that when you got that CR, which happens to me, especially in my young Hodgkin's patients, we do that repeat scan, we don't see anything, right? Duval 0, which means like it's great. I mean, you don't see the hotness. And then I get the question, well, do I need to do this basically 3x? Do I still need to have 4 more left ahead of me? And I'm like, I mean, the guidelines kind of say, yeah, in this case. But it's like, I would love to know that because it is not an easy regimen. And so you all are actually assessing when you get that CR, are the additional necessary?
Exactly.
Potentially.
Yes. Yeah.
And that's amazing. That's less toxicity. Because if I could prove that 2 to 3 or 4 has a high chance of still not having that recurrence, I've spared someone through, a degree of misery. Number 2, it's also the other way around because I have the younger patients, and a lot of times it's the spouse. Understandably, they want their spouse around, etc. When I say it's 4, 2, 6, and they're like, well, what's the determinant? And I'm like, honestly, it's just if you're tolerating it on the labs or you. Again, maximum security, or toxicity. What can I put you through that you can go through? Then a lot of times the patient or the spouse will be like, dude, if they say 4 to 6, I want to go for it, right? Because you don't want to leave on the shelf this chance that should I have gone 6 when you have a recurrence? That's horrible. That's horrible that a non-medical patient has to decide that. Okay. If there was statistical assistance to be able to help us with these decisions, even it's uncomfortable for an oncologist. I'm not asking for empathy, but if you can imagine from my seat, I know in reality this is unnecessary. I know in reality you missing that wedding or that with time of her work very well could be unnecessary. I just don't have the data. I don't have the demonstration, the granular finite metrics, the precise metrics, which is exactly what you're doing. And I think that is something that fundamentally can really spare a lot of economic and financial toxicity, but most importantly, qualitative.
Yeah. So for dogs, it's the same drugs, so CHOP same drugs. Of course, veterinary space does not use the rituximab because it does not work for dogs. So just CHOP instead of R-CHOP. The CHOP for dogs, the protocol is different. So because, as I mentioned, the dog's drug response is very fast, faster than human, and they are tolerate to the drugs better than human. So they actually do the weekly base alternating the drugs. It's not injected at the same time. The prednisone, the steroid, is administered with the other drugs, but the cyclophosphamide, the vancomycin, and doxorubicin, those are actually just injected at weekly basis, not at the same time. So they actually do a more cycle than human. They are going more cycles than human. The reason why doctors want to do this kind of cycle 1 and 2 prediction is that because of this faster reaction and the faster response, if the first cycle does not actually achieve the clinical remission, they just stop there. And they just don't go for the second cycle. But what we observed based on analyzing all the 2,500, all these kind of patients, there are certain portions, pretty good portion of the CHOP treated patients. And then there are a good number of cases that first cycle, it did not achieve a CR, but second cycle, it achieved a CR. And then for those doctors, actually, they just went ahead, even though there was the patient didn't achieve the CR in the first cycle. But there are the doctors just stop there when they see the first cycle, it did not work very well. So
I see.
Yeah, that's why we are trying to do the second cycle. And then if it actually shows that the second cycle will achieve CR, and then the patient is predicted to show a longer progression-free survival. That's definitely please go ahead for the CHOP.
Yeah, no, I mean that's important too because with our diffuse large B-cell lymphomas, myeloma, and acute leukemias, even we get into this uncomfortable or uneasy position of I don't know if you can tolerate the most intense regimen. Because it very well could kill someone literally, or make it to where they can't even tolerate any more therapy. So you're always stuck between this, this thing. Do I push hard because it's a harder, stronger one, at the risk of it all being futile, or do I undertreat and then escalate when I need to? And so, I think in general, if someone's frail, I'll be like, let's just see if we can get away with it because of this concept you're talking about, because we know that sometimes it just like that's adequate what you have. So if it's not a clear indication for a diffuse large B-cell to use what's called escalated therapy, more intense, than R-CHOP, then I'll say, okay, R-CHOP, 2 cycles, reevaluate. But right now, by all guidelines, it's kind of guesswork. It's like 30%, 50% reduction, CR. You feel really good. Obviously, if there's a good response, then it's like, okay, we can consolidate with more. But it's always that case of, I already went the kind of more tolerable way. So am I pulling the trigger early, not to use an intense analogy, but am I pulling the trigger early to escalate when in fact this was the right decision and I can have at least some kind of confidence to say, let's go ahead and do the other 2 because I expect a now logarithmic. It's not a linear, continue additive, because that would be underachieving. But if I could have a little crystal ball and say it's gonna actually still work out, that once again puts an oncologist in less of an uncertain, decision where you just don't really know. But 2, most importantly, and always most importantly, it puts the patient and the family in a less uncomfortable position about a decision. Because me and my patients, we talk it out. We're like, what do we want to do? Do we want to do this? And in general, my philosophy is like, we're all going to pass away. We all have a time, and it's just scary to think about sometimes. So we just have no idea what that year date is. So one of the best things we could do though is to mitigate and minimize the amount of potential regret, and not just in the patient, but especially the family members. That's how I frame everything. Yeah. What is the decision that we're going to, I don't want to leave someone with an injury of regret for their spouse or their daughter or their child. And in a weird way, when you think like that, it makes decisions easier. But it would be great if the decisions weren't so obtuse and hazy and miragey. If we had more granular details, this high degree of discomfort and a highly understated and not talked about reality of cancer management could be, significantly improved upon. And that is what made me very excited about having you and all of this that you're doing, because you're trying to tackle just that. It's not just, we can make sure you get the right drugs. There's a day, I believe, in our near future, honestly, I want to say within 5 years, and maybe that's very bold, that it's not always the most toxic one that's going to be the best. It's not always the most you can tolerate. Let's just give it to you, kind of like back in the day, we would do IL-2 therapy for melanoma and stuff. But it is really a matter of the sensitivity, and people deserve that. And also this kind of I don't want to say game, but it feels like that sometimes. Do we keep going? Do we pass on this time? Do we do the 2 more? Do we escalate? So that's really neat. Do y'all look at, I meant to ask, because I saw y'all look at other drugs. I assume some of them may be non-cancer drugs, because we're learning that a lot of, non-cancer drugs still have an effect on cancer types. Do y'all also look at nutrition or anything like that? That would be a harder, measurement, I guess. Vitamin D levels, kind of all the other variables that I'm hoping machine learning will kind of crack one day.
Yeah, that's what we really want to figure out, but not yet. Our on our test platform, we can add those kind of vitamins there, but the doses and all these kind of things will be matter. We have not tried, except for we have not tried beyond the cell killing, cytotoxic reagents. We are testing the antibodies, but I mean, we have not tested the non-cancer drugs yet.
Right. But it takes these things to then go there, because if you're okay, we've recognized all of these properties, these antibodies and these markers or whatever the case may be. And then when the machine's I'm just going to say yo again, I don't know why now these 2 people responded differently. I've already whittled it down for you 7 layers. Yeah. Now you're at a much more, granular kind of well-defined bucket than it was previously, but there's still something accounting there. And that's why this builds then on, let's look at the other things. Were they on metformin? Were they on a statin? What was their vitamin D like? What was the color of their skin? So all necessary. Again, all stresses why without data, we are just walking in quicksand. I mean, not quicksand, but just drudgery, just thick molasses, because you can't make conclusions and look at the next thing without having, the basics first.
Right.
One thing I saw too, and this is a big thing that pains me, like obviously a lot of things, that's why I have all this white hair in this field for only 5 years full-time practicing, but isn't there something better than a CA 19.9? Isn't there something better than a CEA, like CA 19.9 for pancreatic or 125 for ovarian? And then especially my blood cancer patients, this one hurts me. So we're just going to get a CBC, a complete blood count, and an LDH and a uric acid and kind of just hope for the best. We don't even have intervals to image other than just randomly 3 to 6 months. This kind of conversation again about the surveillance and survivorship phase. One, we hope that there's better intervals because you can tell longitudinally what features make a recurrence more likely at this time frame than the other and then be ahead of it. But two, I saw something about novel biomarkers.
Yeah.
Is it also trying to learn in the background of the background? Are there just kind of features that we could test for either in the blood or on the tissue itself that can help us have more than that kind of 50 years old cytogenetics, staining on a slide, seeing how it dries and blah, blah.
Yeah. This is very important. And then that's one of our very exciting findings, actually. For example, for when we built the human multiple myeloma AI models. So what we tried is trying to collect all the medical charts, the data, including CBC and then fish data and all this together. Our baseline hypothesis is that it should not be just like 1 or 2 biomarkers. Cancer is a systematic disease, so we have to collect as much information as possible and digest in the right way. So that kind of digestion, we just hey, computer, machine learning, you can do this better than me. So we just put those, all these inputs into the black box.
Anything you can give it, just tell us what the relationships are.
Right, yeah. But the important thing is that better biomarker is definitely very, very important. But not only just that biomarker, but combination with the other biomarkers can do better prediction. So we already have many measurements. And then not just we have to figure this is our our team's strengths that we can do this engineering the features. We already have the same data compared to the other researchers, but we can actually find out the interactions between these biomarkers and then try to combine and then detach it and then put that into the black box before we actually put that into the black box. So that's kind of the way that we can actually improve with a limited number of measurements before we actually have a new biomarker. But still, I think that one biomarker cannot do the entire prediction. So I think the combination and how we can digest it is very important.
Yeah, for sure. So in pre-med, this is what really started with this kind of aha moment and saying, wow, this is a problem. And then you had a neighbor or whoever was the vet person whose wife was a vet.
Yeah.
These are tools, you consider them tools. And I love how you said that. Can we just make the job easier? And can we reduce the guesswork involved, which I believe is to an embarrassing degree? For 2024 with the tech that we have in Palo Alto and the golden fields that we hope to migrate to one day in retirement at the age of 90 years old. So one thing I saw when you're looking at machine learning and combinations and everything else was this concept of drug development. Is that in the sense of, identifying things that are maybe resistant mechanisms or escape mechanisms for drugs to then say, hey, here you go. This is one thing we found. And, hopefully you can find something to attack this Achilles heel because it seems to always be deflecting all the treatment we have today. Is that to the capacity that you all are doing it? Or is it even using kind of AI alpha code, engineering to even suggest what kind of, proteins or antibodies may be able to be effective in this?
Oh my God. I love this question because that's another kind of things that we can do. So everything that we talk about is for current patients who really need a treatment right now. Your question is about the future patients, how we can actually help the drug development process using our platform. yes, we can do it because pharma companies love our patient live cell-based drug efficacy test platform because we can test their new drug compound on their target patients' live cancer cells before the clinical trial. So we already provide 3 pharma companies with this CRO service. So one company provides us 2 final candidates, all the cell line results and then mice results and toxicity results. Those are very comparable. Then which one do you want to go for the clinical trial to put the millions of dollars? You have to pick one.
Wow.
But all the data are very similar. So they actually met me at one of the conference, and then they came to our booth, and then I didn't talk about the live, the CRO service, but we have been preparing for that. And then the oncology head just asked us that, I love your live cell concept. Can you actually test our 2 final candidates on the lymphoma patients' cancer cells? And then I just pull up our slides that preliminary data, and then we actually did the service, and the one drug works better than the other one because patients naturally occurring cancer patients' live cancer cells, they behave completely different from mice and completely different from cell lines. And then we can test bunch of same disease, all different patients, who are the good responder to this drug and who are the non-responder to this drug. So that company is very happy with the result because they can select better one, and then they recognize that who are the responder group profile. So they can actually refine the patient recruitment, the inclusion criteria for the clinical trial to make their clinical trial more successful.
Well, more successful, but really what the reason they're not successful is because some patients don't get benefits, and you have to discover that in the trial. That's how we got to where we are. So what you're trying to do is take out those that may end up in another uncomfortable position. Do we go with a second or third line proven drug that has 30% chance of working, or this one that looks really good? You're trying to take out that step for patients to say hey, at least if we can refine it a little further, then that would be great. Now you're subsidizing that with the fact that says pharma, yes, you have to go through these 2 things. It's obviously a huge economic burden, which, by the way, you can listen to the podcast with Greg Simon, the first director of Cancer Moonshot for the White House that we had about a year ago. We are paying those costs for what they pay to get this thing into development in this country, which is why things are so expensive. When I learned that, I was like, that's insane. So even that could ultimately, hopefully, reduce our cost of drug or really health insurance premiums. So that's very interesting. You have a theoretical, you have these drugs that get developed and already have spent millions and millions and millions, but that thing that you're looking at, let's go ahead and now and include the tumor microenvironment and the immune cells and everything, which is what you're doing in a trial in vivo anyway, putting in the body, but let's do it on paper first. Then, of course, you have your toxicity and tolerance, but at least you can concentrate the people that have on a cellular level a chance of having a response. Now that's pretty novel. You've done good, Dr. Lin. You've done good with that one. That one seems
with my team. I'm just representing what we did.
Well, is there anything else? And there's a ton else, but anything else that you're excited about that you want on our minds, if an oncologist is listening or a patient, to just kind of keep in the back of their mind, to help others when the community that may be going through something, or be optimistic about where we're headed?
Yeah, I just want to reemphasize that we are helper. We are not decision makers, and then we need all the oncologists' help, starting from the development up to actual implementation. So we really want the oncologists open their minds to work with the companies like us, other companies also. And then precision medicine, we witness that it is true. I think all the clinicians already know that every patient is different. And then every dog is different too, and cats too. So we all know that precision medicine is the way to go. And then I hope more precision medicine companies will emerge, and then more collaboration between hospitals and then academic institutions and then industry people all together to make up patients the treatment improvement better. And the second thing is that for hopefully the patients can understand that this is a prediction. This cannot be 100% correct. So we all do our best, working with oncologists do their best, and then companies do our best. But please understand that it's not perfect.
Yeah, there will always be guesswork, but you're trying to make the guesswork less.
Yeah, and then the third thing is that it's as you emphasize, it's very important for the other precision medicine companies that don't try to change the clinician's behavior. We are not trying to provide a new combination or like which did not go through the clinical trial yet. What we really want to do is that there are multiple treatment regimens that is being commonly used, and which one these clinicians can choose. I think that's the better way to start. Of course, later, if the clinicians wants to test the new synergistic combination, we can actually do that. But it's technically possible. In the clinical application, we have to start with what clinicians are doing right now, and then make the decision better. I think those three are things that I want to emphasize.
Dr. Seung-Won Lim, I just want to say thank you very much for everything you're doing, your passion, and I'm just eager to see what you bring us in the oncology community.
Thank you very much. Thanks for having me. And then I really enjoyed our conversation. Your questions were excellent, and then I was so excited to answer those questions. Thanks so much.
I get it. Yeah, I love it.